Act 1. The Discovery of MSCs. Alexander Maximov was born in 1874 in St. Petersburg, Russia, to a merchant family. He received an excellent education and entered the Imperial Military Medical Academy. He continued his academic excellence and showed interest in experimental medicine, graduating as the top student of his class in 1896. He was nominated to be the professor at the Department of Histology and Embryology at the Academy, where he held the position until 1922. Among the scientists who nominated Maximov was Ivan Pavlov, known for Pavlov conditioning, after his famous experiment conditioning dogs with food and ringing bells. However, after the Communist Revolution in 1917, Maximov immigrated to the US with his wife in 1922, where he became a professor at Chicago University. Maximov introduced the theory of hematopoiesis, where a type of cell in the blood can transform into different connective tissue cells like fibroblasts. His common origin theory for erythrocytes, granulocytes, and lymphocytes was not accepted at the time, as it was difficult to prove. In his articles, Maximov hypothesized that a lymphocyte-like cell acts as a common stem cell and migrates through tissues to multiply in appropriate environments. Maximov passed away in 1928, unable to prove his theory of an origin cell. However, Dr. William Bloom, who worked closely with Maximov, finished his textbook of histology in 1930, becoming the standard text for medical students. A few years before Maximov's death, a young boy named Alexander Friedenstein was born in 1924 in the former Soviet Union in Kiev. Friedenstein's family moved to Moscow in 1928, where he graduated with a degree in medicine in 1946 and a PhD in skeletal regeneration in 1950. While the Cold War was in full swing, Alexander Friedenstein worked on stromal cells while being isolated from the larger Western scientific community. Instead of working on blood stem cells, he asked the simple question of how blood cells are born. If the bone marrow makes blood cells, what kind of environment determines what they become? This propelled him towards a search for a scaffold, or stroma. Friedenstein idolized Alexander Maximov, publishing articles on stromal precursor cells in the 1960s and 70s. He continued the work of Maximov by proving Maximov's hypothesis that origin cells exist and play a role in hematopoiesis, mainly by transferring a hematopoietic microenvironment (HME). He developed a simple test, a fibroblast colony forming unit (FCFU) assay. By taking bone marrow cells and culturing them in vitro, he identified certain cells that multiplied and transplanted them into animals. After transplanting FCFU into the kidney capsule of a mouse, he found that the cells built a fully organized organ, complete with the bone and bone marrow inside. This led to the discovery of a type of cell that can produce a HME, hematopoietic microenvironment. This is what we call today as mesenchymal stem cell, but we will get back to this later. Friedenstein then defined them as stromal precursor cells that are, number one, highly proliferative and clonogenic in vitro in a petri dish, and number two, capable of self-renewal and multilineage differentiation in vivo in the body. He demonstrated that single-cell derived clones of stromal cells can differentiate into bone, cartilage, and connective tissues in vivo. This achievement propelled future cell therapies and tissue engineering, which is growing exponentially in the present day. Friedenstein's research accomplished one of his goals, which was to restore Maximov's reputation, after having received skepticism for his radical theory of a common origin for blood cells. Unfortunately, because Friedenstein's work was done in the Soviet Union, it took a long time for his discovery to reach Western scientists. With a collaboration with Dr. Maureen Owen, Friedenstein's articles on stromal precursor cells were disseminated worldwide, and he is now recognized as the person who discovered mesenchymal stem cells. Dr. Owen's work helped to promote widespread interest for stromal precursor cells. This culminated in Dr. Arnold Kaplan naming the stromal precursor cells as mesenchymal stem cells, MSC, in 1991. This is still the name that we are using now. Arnold Kaplan was a professor in the US and looked at Friedenstein's stromal precursor cells. Because he was an expert in skeletons, he thought that these cells don't just build a hematopoietic microenvironment for blood cells, they are the ancestors of all skeletal tissue. This was a very strong rebranding. When you call the cell a stem cell, it shifts the focus of the field from hematology to the field of tissue engineering. Kaplan's motivation was to unlock MSC's regenerative potential for bone and cartilage repair. He made the scientific world pay more attention to MSCs. However, the full potential of this cell has not yet been revealed. It will take another 2 decades of work to uncover the full range of applications for MSCs. Act 2. The Immunomodulation Paradigm Shift. The first big surprise happened in 2002. Dr. Marilyn Bartholomew was testing MSCs in animal models for regenerative purposes, but she noticed something strange. When she infused monkeys with MSCs, the monkeys were not just healing bones, their immune responses were also dramatically suppressed. Bartholomew's key 2002 study showed that, in baboons, human MSCs could suppress the immune system long enough to prolong the survival of skin grafts. This was massive and unexpected. We have discovered that MSCs were also powerful immune modulators. The table on the slide shows that baboons receiving MSCs had extended donor graft survival up to 13 days compared to control of 7 days, illustrating MSC-induced donor-specific immune suppression. The field now on MSCs was turned on its head. They were not just for tissue repair, but they were a potential therapy for immunological diseases, such as graft versus host disease, Crohn's disease, type 1 diabetes, and multiple sclerosis. This paved the way for research in clinical applications, which began shortly after. But one major question remained: how were the cells doing this? Scientists assumed that donor MSCs engrafted and became part of the recipient's tissue, but this was not the case. In 2009, a second, even bigger surprise came from researcher Li Ranghua. She was using MSCs to treat heart attacks in mice. They treated the mice with MSCs, but they couldn't find MSCs in the repaired heart tissue. They had disappeared. However, the heart was still healing after the disappearance of MSCs. The heart slices on the table in the slide demonstrate a visibly smaller pale area in MSC-treated groups compared to untreated knockout TSG6 knockout groups, visually confirming that MSCs improve cardiac repair through TSG6-dependent paracrine mechanisms. This was paradoxical. Li found a new idea for MSCs. MSCs could work like temporary drugstores. They rush to the site of injury, release anti-inflammatory signals included in their paracrine secretum, and then die off. The therapeutic effect comes from these secreted factors, not from the cells themselves. This was a true paradigm shift from differentiation to secretion. This helped to explain a lot on the mechanisms of MSCs, but it also created a new problem. If MSCs are drugstores, why do clinical trial results vary so much? By now, many trials have been started and are undergoing, but results have been varied. Sometimes they suppress immunity, but other times they seem to have no effect, or even and opposite stimulatory effect. The final puzzle was addressed by scientist Ruth Waterman in 2010. She proposed the license to kill model. Think of an MSC not as a simple on and off switch for the immune system, but as a dynamic cell responding to its environment in order to activate its effects. And these effects will change each time depending on the environment. Waterman showed that the inflammatory environment itself licenses or polarizes the MSC. A signal like IFN gamma from T cells could push it into an anti-inflammatory state she calls MSC2, but a different signal, like through another pathway with TLR4, could make it pro-inflammatory, which she called MSC1. Microscopy images stained for adipogenesis and osteogenesis shows that TLR4 primed MSCs favor osteogenic differentiation while suppressing adipogenesis, whereas TLR3 primed MSCs inhibit both, highlighting how innate immune signals polarizes the MSC. This was the final piece of the immunomodulation puzzle. It meant that MSCs have a dynamic immune response. Its actions depend on the signals it receives from its environment, and this explained the variability in clinical trials and taught us that we couldn't just inject cells. We have to understand and precondition them for the specific disease environment. So, in just a few decades, we went from Friedenstein's discovery of a tissue building cell to Kaplan renaming and framing the cell as a stem cell to our discovery of its immunomodulatory effect. At the same time, numerous trials have begun to research the potential of MSC-based treatments, including in China. Act 3. Bench to Bedside. In 2004, Dr. Katarina LeBlanc in Sweden faced a child patient with graft versus host disease of the gut and liver, a type of immunological disease that occurs after stem cell transplantation. No treatment worked, and faced with no other options, Dr. LeBlanc infused donor-derived mesenchymal stem cells into this single critically ill child. It was a Hail Mary pass based on Bartholomew's 2002 data in monkeys. The result was almost outstanding. GVHD symptoms improved dramatically, and the report was published in 2004 in The Lancet, one of the top medical journals in the world. It was the first clear signal in humans that MSCs could be a powerful rescue therapy for steroid refractory GVHD and perhaps other immunological diseases. Clinical trials began immediately after, with Korea being the first country to approve MSC-based therapies in 2010. MSC therapies were difficult to regulate due to the product being live cells and were adopted slowly. Only 20 years later, in 2024, the US FDA approved America's first MSC-based product, Ryoncil, for pediatric acute graft versus host disease based on phase 3 data conducted by Dr. Kurtzberg. Almost in parallel, a team in China was working on the same indication. They conducted their own phase 2 clinical trial for adult steroid refractory acute graft versus host disease, AGVHD. In 2025, China's NMPA approved Reibosheng, the first MSC product for adult AGVHD in China. This is also the first stem cell therapy approved in China. Today, there are 15 MSC-based therapies, and the global market for MSCs is growing rapidly, with hundreds of trials underway, and for other indications like multiple sclerosis or type 1 diabetes. You may notice in the table that the first 4 MSC-based therapies were approved in Korea. This is because the Korean government provided its regenerative medicine industry with R&D funding and fast-track reviews, approving therapies with small-scale phase 2 data with potential efficacy, encouraging local companies to commercialize quickly. However, the FDA and the Chinese NMPA are stricter and requires phase 3 randomized controlled trials. These regulatory bodies requires proven efficacy instead of Korea's potential efficacy. In general, predefined clinical criteria is difficult for MSCs, especially for immunological diseases, due to patient biology, MSC heterogeneity, and differences in procurement. The ISCT defines MSC with a minimum criteria in 2006, but since then there has been no change. It is still hard to understand the mechanisms of how the variability of MSC impacts their immunomodulatory and homing capability. We are still trying to understand the best ways to use MSCs in terms of administration, dosage, cell quality, and in vivo tracking. To sum up, more than 100 years after the introduction of Maximov's common origin theory, the hematopoietic stem cell, it formed the new basis for treatment strategies involving hematopoietic stem cell transplantation and for immunotherapies. This work of Masimov was continued on by Alexander Friedenstein in Russia, who proved that the cell existed and saw it as a scaffold cell for building tissue. The cell was brought to the West and further studied by Dr. Maureen Owens at Oxford University, and it was rebranded by Dr. Arnold Kaplan, and both scientists saw its regenerative potential for bones instead of hematology. The paradigm for MSCs finally changed when Dr. Bartholomew discovered its immunogenic properties, and Dr. Lee revealed that it worked through cell-to-cell paracrine signaling. And finally, Dr. Waterman revealed that the signals can be controlled by its environment, and henceforth we can modify MSCs. Finally, the clinicians translated this science into medicine, turning a biological quick curiosity into a life-saving treatment for immunological diseases. MSC's role in immunology has been a serendipitous discovery, and it shows that basic research can become life-saving drugs by scientists passing the baton across generations and collaborating internationally. Thank you for listening.